Okay, so going through the study guide first, it said give an example of an herbivore, omnivore, and a carnivore. So obviously, hopefully you remember that from nutrition, but slide one, herbivores, which their diet is primarily plant. Cattle are the ruminants, and then horses would be the non-ruminants, and then the carnivores are cats, which are the true carnivore, and then humans, pigs, and dogs. Primarily, I focused on dogs eating both. Give an example of mechanical digestion, chemical digestion, and a place where each occur. So when we talk about digestion, if something mechanical is usually movement. So in the mouth, that's mechanical. Stomach, basically almost every part of the digestive tract does have mechanical digestion, but not every place in the GI tract has chemical. So chemical, the mouth would be one place because you have chewing, but it also, that's where saliva is deposited, and that begins chemical digestion. Stomach has both, stomach acid and stomach contractions. The only place really, the esophagus, is really just kind of moves the contents from the mouth to the stomach. And that's primarily movements. There's not a lot of digestion that's occurring because of the esophagus. So for the most part, small intestine, that's where digestive enzymes are released, and that's where absorption. He got the lid for the microwave. Thank you, AJ, for bringing that to me. Okay, we're gonna put that up here. Okay. Buddy, you were so proud that you got that. Okay, so just being familiar with locations and what the difference between chemical and mechanical digestion. Identify structures within the GI tract and outside the GI tract. So if you remember, the mouth connecting to the esophagus, stomach, small, large intestine are all the, he's quite entertaining, Beth. He's figured out he can reach. He was like, oh, Mom, it's your birthday. Let me give you the microwave lid. Oh, thank you. But yesterday, I caught him. He's just like so tall now. He was reaching way back on the counter and trying to get a spoon, and he's pretty good at it. So I have to now watch what I leave on the counter. Yeah, there you are. You saying hi? He's a good boy. Okay, so those are structures within the GI tract, but the outside of the GI tract, I talked about the pancreas and the liver. And so they play an important role in digestion, except they're not within the GI tract. So they are outside of that GI tract. What are the primary structures of the tooth? So the basic tooth, this is the picture. Again, there's not going to be a picture of this on the test where you have to label a tooth, but just be familiar with, the crown is what you can visibly see. The root is inside of, the bone of either the maxilla or the mandible. And then the center is the dentin. The pulp is the sensitive structures. What's the difference between the surfaces? So you've got the four common surfaces. Your buccal is any tooth that can touch the cheek. Now the teeth on the top, and usually we're talking about the canine back. So the canine has a buccal surface because the canine does communicate to the cheek. And that can again be top or bottom. So buccal does not tell me, it just tells me that it's an outside surface. And labial is the outside surface, but this would be pertaining to the incisors because this is communicating with the lip. So again, labial can be top of the jaw or bottom. And then you've got lingual, which is the inner surface, but that's the bottom teeth because they touch the lingual is tongue. So they touch the tongue. And then palatal is pertaining to the upper teeth because the inside touches the palate. Okay, so palatal and lingual, that will tell you if it's a top, a tooth on the top or tooth on the bottom, but the other two, it could be top or bottom. And then it says for number 6, what's the difference between a brachiodont, hypsodont tooth, and one species that has each? So the brachiodont, and think about a short growing period. Like when an adult tooth, brachy means short. Can also, so that's do not grow continually. So carnivores and people, ruminant incisors, but primarily dogs and cats, they get their adult teeth, and once they emerge, if they lose that tooth, it's gone. It's not going to regrow. Now a hypsodont teeth would be like horse incisors and their cheek, but you've got lagomorphs, which are rabbits, rodents, and the canine teeth of a boar, and these are a continuously growing tooth. So they either, we talked about teeth floating in horses, which is actually they will get the tips filed off. If you look at this picture, whoopsie, they almost look like they've got these kind of like ridges. If an animal, now normally when they translate their jaw and they grind like their grain or grasses, that'll help file, but sometimes if they eat more grain, less forages, and also like rodents and rabbits, a lot of times you can buy them little wood blocks or things to gnaw on, and that helps file them down. But even, like a rodent or a rabbit, sometimes they do have to get their teeth trimmed, because maybe they're not using them properly. Maybe they don't have the things that they need to chew on. But they will be continuously growing. So the way that that's controlled is by them chewing things or, doing something to help file them down naturally. And now I didn't ask a specific question about this, but radicular hypsodont versus aradicular hypsodont is, is whether the root is remaining closed or open, but don't worry about that for the test. And then deciduous, I don't have a question about it, but deciduous teeth are baby teeth. So do know that deciduous means baby teeth. You did need to know the 4 names, and even though I didn't ask a question on the study guide, I did tell you, and maybe you want to make a note, to know the dental formula for the canine adult and the bovine adult. And you need to know what each letter stands for and the order too. You don't want to put MCPI. It goes incisor, canine, premolar, molar, and then how many. Now, the number in front is the top jaw, which is the numerator. The number on the bottom, which is the denominator, is the bottom jaw. And this formula is half of the mouth. So, you'll get, when you add that up, you get half of the total number. But again, it only represents half of the mouth. You didn't need to know the Trident system. You'll go over that in Animal Tech 4. What are the functions of the tongue? So it is to move food into the oral cavity and for grooming. It does help with, temperature sensation, pain sensation, also thermoregulatory. When an animal's panting, they look like they have the biggest tongue in the world, but they can help, by evaporation and lose some body fluids through panting that will help cool them down. Where is saliva produced? What is its function? So it's deposited in salivary glands. But it's really, this is also, I didn't ask a specific question on what are parts of saliva, but it has quite a bit of things in it. I think the most important, it is composed of water, but it can change from watery to viscous. Sometimes animals, the saliva is very sticky. Sometimes it's a little more drippy, more watery, because of the glycoproteins. But it does have salivary bicarb in it. Bicarb is a buffer that will help buffer the contents before it moves into the GI tract. And also amylase. And that is where carbohydrate digestion begins. But again, I don't want to, no, don't tell me on a test, saliva is not produced in the mouth, but the salivary glands are exocrine glands. And let me talk about the next slide here. So here's all the functions. So if I said, what are the what are functions, you could tell me, you know, lubrication, antibacterial. pH regulation is the bicarbonate. So bicarbonate helps regulate pH. Acidity versus alkalinity is helped maintain. Now, enzymatic digestion, and it's, you know, these are three main salivary glands that do produce, and again, they have a duct or a tube, and that tube connects into the oral cavity. So when they start to chew and eat, and those glands constrict and kind of squirt or deposit some of the saliva in the mouth. Let me, okay, I talked about, and I know we talked about the pharynx before, you know, it's the common passageway, but that's where food will go from the oral cavity, move to the back of the pharynx, and then move into the esophagus. And the esophagus, it says, what is the function of the esophagus? And it's really to move food from the oral cavity to the stomach. It's a tube that connects. And well, I'm going to come back to that in a second. And then peristalsis is muscle contraction that propels food. It's a wave-like contraction. So peristalsis, if you see what I'm doing with my hand, it's kind of like a wave. So now remember, there are two layers to the muscular layers to the esophagus and the intestines. So you have the longitudinal fibers, which long waves, and then you have circular fibers, which help segment and mix. So both of those muscles are autonomic. Now let me go back. I don't have a question about this, but if you remember, from the esophagus down is autonomic control. You know, movement of the GI tract is going to be controlled by the autonomic nervous system. When they're in parasympathetic stimulation, the GI tract is moving. When they're in sympathetic stimulation, GI function slows down because the resources are needed elsewhere. So they kind of take the blood flow and say, all right, I'm going to shift it over here, and therefore activity slows down. So from the time, now, when food is in the mouth, it's under like a conscious control because, you know, if you've eaten something, sometimes you have to chew something longer and sometimes you don't. I mean, you can chew, chew, chew. Once it does move back through the pharynx into the esophagus, then it's under this autonomic control. So it's under an automatic control. So let me go back for a second. It says, what do the terms prehension, deglutition, mastication mean? So prehension, which is grabbing, mastication is chewing. We get salivary secretions, you know, which the nervous system is going to secrete saliva when something touches the mouth. But remember, some of the salivary secretions can also be that's the cephalic phase of digestion. It's just the anticipation of a meal can get saliva secreted. But then when it moves into swallowing to the esophagus, that's called deglutition. So do be familiar with these terms. What are the functions of the stomach and what is chyme? So the stomach, first of all, it's storage. The monogastric stomach, serves as storage. But it has mechanical and chemical digestion based on stomach contractions and secretions. But it also produces a factor for B12 absorption in the intestine. So when the factor is secreted, it will cause B12 to be absorbed in the intestine. Chyme is the food in a semi-liquid form. So when food enters the stomach, it's called a bolus. So when they chew food, a chunk of food enters the stomach, it's a bolus. As it's digested, and when it leaves the stomach, it's now called chyme. And that chyme is a semi-liquid consistency. Now, eventually, when chyme gets to the large intestine, that's where it becomes feces. The water component finally kind of gets absorbed, it solidifies and becomes solid fecal material. So it kind of goes from food in the mouth, bolus in the esophagus, chyme leaving the stomach, and eventually, no, you can't have that, and eventually becomes feces. So you can't have your microwave thing. No microwave thing. Okay. Okay. Don't be mad at me. Okay. It says describe the anatomy. Oh, and just to review, the two. I literally think he's gonna climb on the desk. No, get over there. No, no, no. You can't have it. No, go sit. Go lay down. Sorry. Um, so you know, like if you get, so like when you want to microwave something. And I microwaved something and I had put this out on the counter. I didn't use it. And then all of a sudden I heard him and he just carries it in me, so proud. Like he loves chewing on plastic. Like, yes, he's very 9 months old right now. Now he's trying to lay on top of Bo. This is funny. But yeah, no, he wants to help teach. That's good. We can talk about your stomach and all the stuff you've eaten. I can, yeah. This is funny, very puppy. So yes, that's a microwave lid. Just to remember, in dogs and cats and horses, we talk about the monogastric, meaning one stomach, and then ruminants, sheep, goats, and cattle, have a 4-chamber stomach. Some people call them, and a lot of people say if they have 4 stomachs, like a number 4. It's not like they're 4 separate structures. It is one structure with 4 chambers. So, you know, I mean, it's like I said, a lot of people just call them 4 stomachs, which is a very common way, but those are what, you know, a ruminant. So there is a difference between, you know, there's a if I say, give me the name of a monogastric species versus a ruminant, then that's the difference that I'm talking about. The anatomy has these kind of compartments. So I do want you to know the esophagus empties into the cardia. That's where the stomach or the esophagus enters the stomach. And then it enters the body, which is kind of the middle. It has this pocket up at the top called the fundus. And this is what can really stretch. So when I showed you that video of the Shih Tzu that ate all the, you know, rubber bands and underwear, a lot of that was probably getting shoved up in that fundus, and that fundus can stretch and stretch and stretch and stretch, and it can kind of just keep getting shoved up. Now, the end of the stomach, there is this pyloric antrum or the pylorus, and that area gets narrow and thicker. Now, it's thicker because the muscle is thicker, because once contents move to the pylorus, that's the pylorus, it starts to pump. And so that's why it's thicker. And then it has, the pylorus is a circular opening into the small intestine. There's a muscle there called the pyloric sphincter. And that sphincter will open and close. And as things get closer to the pylorus, muscle contractions increase, and that pumps contents out. And I did caution, you know, this is a little more narrow than the cardia. So unfortunately, this can be a spot where we can potentially get some foreign bodies. Are you listening, Boxer? They eat stuff, you hope that it could potentially pass out. So the pylorus is the actual narrowed opening, and the pyloric antrum is the space before. So this whole space is the antrum, and then this narrowed area is the actual pylorus. So yeah, so that when they refer to the pylorus, it's the opening, and there's a sphincter. So sometimes people just call it the pylorus. Sometimes people call it the pyloric sphincter, but they're really talking about the same spot. There's a muscle. The problem is it only stretches so big. You know, this part of the stomach, the fundus in the body can kind of stretch. The pylorus is that pyloric antrum and sphincter area aren't going to stretch that much. It's thick, much thicker muscle. And then the rugae, I didn't ask a question about that, but the folds on the inside. And then I'm going to talk about pepsin, but inside the wall of the stomach are the gastric pits. So when we refer to the monogastric stomach, we call it glandular because it's secreting. And the glands that are secreting, or these cells, live within the stomach wall. So when something hits the stomach, when food, the bolus enters the stomach, there's going to be a stimulus and they're going to secrete. Obviously, we've got mucus cells, parietal cells. But let me talk about, it says 14, what's pepsin and its purpose. So pepsin is a proteolytic enzyme, and it's secreted by these chief cells. So the chief cells are going to secrete, let me back up. They're going to secrete pepsinogen, and that is inactive. Pepsinogen is inactive, but when it's secreted into the stomach environment where there's hydrochloric acid, it's converted into pepsin. Pepsinogen and then remember my little helpful tip for you guys, anytime you see gen on the end of it, it's typically inactive, and it's going to generate the active form. So if gen or generate helps you remember, when you see gen on the end of it, then it's going to be activated. Like there's trypsinogen becomes trypsin. Chymotrypsinogen becomes chymotrypsin. So when you see gen, it's typically something that's secreted and active, and then it's inactivated once it enters the environment of like the small intestine or the stomach. In this case, hydrochloric acid of the stomach. And remember I said this is proteolytic, so this is going to help digest protein, like lytic, breaking up protein. And I like a Pepsi with my protein. I don't know if that helps you remember. And then it says, what is the difference between cephalic phase and gastric phase? I think I've talked about that. Cephalic phase is anticipation, cephalic pertaining to brain, where gastric phase begins when the food actually hits the stomach. So it's just talking about the time. It's like when a stomach is sitting there with nothing in it, just the beginning of thinking of food can begin to get these secretions going. So it doesn't necessarily mean they've had to have food yet. Let's see. What does each part of, and there's a few phases here. The main thing, and I didn't ask a specific question, but this is talking about the process, and you're not going to have to describe it in this detail, so don't worry about that. There's the cephalic phase it talks about, and the gastric phase. And then gastric motility, it says, I'll come back to number 16 in a second, but how is gastric emptying controlled? There are pacemaker cells. So within the stomach, these special cells, when food hits the stomach, it's going to begin to contract, and it's going to begin to break up particles and mix it with the hydrochloric acid and the pepsin, and it's working on turning it into chyme. So sometimes what happens when there's growling on an empty stomach, sometimes the acid can irritate, and sometimes there's a little bit of gas, and that's causing like a movement to expel the gas. So you kind of feel like, because in all actuality, the stomach is always moving, even though there's not food in it at all times. But sometimes air is going to build up, sometimes you swallow some air, but also gas formation, and it kind of just lets out that, like it's going through a contraction, and that growl is part of the air moving, and some of that fluid creates that sound. So You might hear mine in a minute. So good question. So between the pacemaker cells and then the muscle cells, or the muscle layer, which is it's going to be initiated by acetylcholine. So as the pacemaker cells get stimulated, acetylcholine causes calcium to be released, which initiates the muscle contraction. Um, and that will create the muscle contraction. But again, it can be gastric or cephalic. Now, what each part does, for 16, the body is the primary mixing chamber. So when the bolus comes in, it's going to mix with the secretions primarily in the body. It can move into the fundus, but the fundus is kind of like expanding to kind of, get a higher amount of food intake. Like, sometimes you go to like a buffet or you order too much, your eyes are too big for your stomach kind of thing, and you might take in more food than normal. And that fundus can so if you think about the body mixing, stuff can move in to the fundus and move in and out and get mixed. By the time it reaches the pylorus or the pyloric antrum, then it's pretty much mixed and ready to move on to the intestine. And that's when it begins to pump. But again, things need to move slowly to the pyloric antrum because you don't want to push everything out. Ideally, what happens is it gets pumped in segments to the duodenum so that the intestine can process. So that it, again, you're going to get a little bit pushed out, a little bit pushed out, and, it's going to work its way through. And I talked about gastric emptying. Where is amylase located and let me mention one thing there's not a question about it, but this peristaltic contractions kind of leads back to number 17 of this gastric emptying. The fragmenting of small particles can influence how fast it goes out. So, for example, you may have a dog that inhales their food, like it doesn't even look like they chew. And some dogs do that. Now, when it gets to the stomach, it's going to be a little bit harder to process because it may be more whole. If a dog chews, it's chewed up, it's mixed with saliva, it's going to digest a little bit easier. So sometimes it's related to the movement and the size of the particles, because remember, if something gets pushed to the pylorus, which is that narrow opening, if it thinks it's too big, it will retropulse it or push it back into the stomach, and then the stomach will churn and churn and churn until it's an appropriate size. So sometimes, it's hormones. It's also size of particles that can influence how fast things leave the stomach. Oh, hold on a second. AJ, stop. Come here. Come here. Come inside. Come on. You're staying in here. I don't trust you. You're my aunt now? Okay. Usually he's sleeping right now. I apologize. He's kind of being a bad boy. Okay. So in that Shih Tzu, that's eating all those rubber bands and all of that stuff, if it probably tried to push out the pylorus and then got pushed back, and then so much built up, it kept pushing into that fundus, and it started filling up the fundus. But then it got to a point where then the dog, my computer. Then probably what happened is it got to a point that it started becoming, the dog started showing more clinical signs because the stomach was just then way too full. So that can kind of, and just like this guy got into the trash a couple days back, or it was like last week or a week before, but he vomited. It was like 3 or 4 days later, and then I saw what was in the trash. So that probably was hanging out maybe in his fundus. You know, it didn't get pushed into the intestine, thankfully. Maybe it would have blocked. And sometimes he poops out lots of things, and strings and cloth, and he loves paper towels and things like that. So that's kind of, you know, the other thing about gastric motility is the main thing I want you to know. There's the nerve fibers detect the presence of food, and as food gets closer to that pylorus, then the contractions are going to increase. So that's going to increase it. Gastrin is a hormone produced by the stomach, and that can be adjusted with the presence of food, and that can also increase contractions. Okay. What are you looking? Oh, I said, what is amylase? Oh, I'm sorry. I keep going back to things. Let me just mention, and I'm going to talk about this in a minute, but there's also hormones in the intestine that can control gastric emptying. Remember, CCK and cholecystokinin, which is cholecystokinin and secretin. If the food or if the chyme enters the duodenum, and if it's like very high in fat and very high in protein, then it can actually, basically what will happen is these hormones are released from the duodenum, but will tell the stomach to slow down. Therefore, the intestine can spend more time digesting that material. So even though they're released from the duodenum, it affects the stomach. So, you know, you can see there's various things that kind of control this emptying process. I can't see my questions. Okay. Amylase, I will say for 18, amylase is released from the pancreas and it helps digest carbohydrates. And pepsin is a proteolytic enzyme that breaks down protein. Okay. Says 20. Let me move on to, oops. Oh, no. All right, hold on. My PowerPoint just crashed. Let me see if I can pull it back up. Can you guys see the PowerPoint anymore? No. Amylase, Morgan, is released from the pancreas. Okay, for some reason, let me, all right, sorry, my computer is just hating me today. Okay, you put your lip on my paper. Come here, stop. Let me try this something again. I might have. Hold on, guys. Sorry, my computer is just. AJ, please, thank you. Okay, I see if I get this to work. This is, yeah. Ooh. Wow. Okay. I may have to just verbally, okay, maybe can you guys see this? Okay. This is like a PDF I really apologize. Let's see if I can get this. Ah, there we go. Okay. Okay, let me move through. I want to get through. Okay, so for number 20, sorry about all this. Hopefully my computer's going to make it through. So the four chambers, the first three are the rumen, the reticulum, and the omasum. Which chamber is the true stomach? That is the abomasum. So the abomasum acts like our stomach, and the abomasum empties into the duodenum. So the abomasum is the last chamber of their stomach. And then it says, which chamber is the primary fermentation? That is the rumen. So the rumen ferments. It said, which parts of the ruminant stomach are glandular, non-glandular? The only glandular component is the abomasum. Let me, okay, so the abomasum does have glandular tissue, so that would be more, it would be, but like in the cow, food does not spend a large amount of time there. It pretty, you know, like ours can be storage, but it's going to move through a cow, just kind of pass through the abomasum. But it does have a glandular component. The non-glandular portions is the rumen, reticulum, and omasum. Now they're non-glandular, so they do not secrete stomach acid. So you remember when I talked about the pH of the ruminant, those non-glandular components are going to be like your, you know, upwards of 7.6, 7.8, more alkaline. And then the abomasum would have a pH closer to our pH, which can be as low as 2, not that it would always be 2.0, but could be as low as 2.0. What is the purpose of the esophageal groove? And this allows, when a calf is nursing, it will bypass the reticulorumen, you know, basically bypasses those first compartments, and it goes directly to the omasum. That's because you don't want their milk to ferment, which can lead to a buildup of lactic acid. So that's all that that does. And it says, which part of the stomach has a honeycomb appearance? If you go back, that's the reticulum. Sometimes it's called the honeycomb stomach. And then the rumen, just so you know, it's the primary fermentation chamber. It's the largest. And I do want you to know it is on the left side of the cow. So that's where we, you know, I showed you the video of that calf that was bloated. And the left side was like huge. What is the four-step chewing the cud? And the other thing here is in this rumen reticulum, it's moving back and forth. So there's, you know, a couple days worth of food in there. And, you know, so you've got what was eaten today, what was eaten yesterday, gases, and it just kind of keeps churning and mixing. And basically there's microbes, protozoa that are breaking, you know, there's no stomach acid in here to break this down. So it's basically like a little ecosystem in there. The four-step process is called, you've got, and I want you to know the steps. After they swallow their food initially, they regurgitate it into their mouth. They're going to add saliva, re-insalivate it, then they remasticate it, and then re-swallow it. So sometimes, I guess, if you look at a cow and they go, they kind of look like they do like a little burp. That's them bringing their food back up into their mouth. They chew, re-insalivate it, swallow it, and then and that's part of their digestion process. For 25, this wasn't in the notes, this was the video we watched. What is a fistulated bovine used for? So this is when they have a permanent surgical opening implanted in the rumen. And number one, it's used for teaching purposes. So you can keep, you know, like I said, most veterinary hospitals, maybe high-end bovine facilities, will usually keep a ruminated cow or steer for teaching. That's why we had one at Purdue, but it will also can treat other sick cows. So you can access, like I called it Activia for cows, where people take probiotics. You can take the protozoa fungi, because a lot of times when animals are sick in the hospital, their GI bacteria are thrown off and it affects their ability to digest. So we do what's called a rumen cocktail. We take some of that fluid and contents out. We would sometimes put it in a blender. That's why we kind of call it a cocktail. And then we could tube it to, and it's called transfoamation. So to transfoamate, we take this contents and give it to another one, and it will re-inoculate their GI tract with the proper bacteria, GI flora. What are parts to the small intestine? What is the mesentery? So it's DJI, DJ ileum. What the word DJ? Duodenum, jejunum, ileum. If you look at the three parts, the duodenum is the first part of the small intestine, and the pyloric sphincter attaches to the duodenum. The jejunum is the longest part, and that's where you have the majority of the digestion. And then the last part is the ileum. And remember, let's see if I have a question about it. Oh, the mesentery is the connective tissue in the blood supply to the small intestine. So I kind of showed you those pictures. You got the intestine. It kind of looks like almost clear see-through, and then you see like blood vessels. And that's the mesentery. Where does most chemical digestion occur in the small intestine for 27? That is the jejunum. And what are plications and villi? So plications are folds. So sometimes if you look at pictures of the intestines, it looks like it's kind of like folded in accordion. Well, you never want it to really be accordion. That's a problem. But the folds are plications. So it allows for an increase in surface area for absorption because those folds can stretch. And then the villi, an intestinal villus, this is a villi. It does have a microvilli border, but this one is a villi, and then it's covered with this brush border. But this helps with the surface area for absorption. You remember I tried to draw that picture. So they are, the best way to answer it, they are adaptations within the small intestine that allow for increased surface area and absorption. That's kind of the best way. And then in each villi, artery, vein, lymphatic vessel. The lymphatic absorbs the fat and the lacteals, and then your blood capillaries are going to absorb the nutrients. And then these were the secretions that I talked about earlier that come from the duodenum, which if it detects fat or acidic chyme, it releases bicarb and increases, so basically what these do is they will, they can decrease, secretin's going to decrease stomach acid because, hey, it's too acidic, or it's going to increase bicarbonate or digestive enzymes because it's too fatty or, protein, and that will slow down the stomach. It gives the duodenum more time to process or begin digestion. That's kind of the theory is, hey, don't keep sending me stuff from the stomach. I'm trying to digest what I've got. It's either too acidic, too fatty, too high in protein, takes longer to digest. These slow down the stomach and give the intestine more time to digest. Let's see here. Oh, pancreas is located around the bend of the stomach and the duodenum. It's in the mesentery, so it's attached to that. This is the mesentery, and then it's attached here. It's like a flat organ, but it's right around the bend of the stomach and duodenum. What is the exocrine function of the pancreas? What's the endocrine function? Endocrine portion is where we have our pancreatic insulin or islets, and they produce insulin and glucagon. And the endocrine pancreas secretes within, no duct, secretes within the blood. The exocrine pancreas has a duct that enters into the duodenum, and that's where it secretes, or it's going to excrete bicarbonate and digestive enzymes. So that's the difference between the exocrine function and which one has a duct, the exocrine pancreas. Where does the pancreas deposits its digestive enzymes? Into the duodenum, which again, amylase, lipase, and then there's proenzymes, which are secreted inactive, which is trypsinogen. So I talked about exocrine pancreatic insufficiency, and when they don't produce enough. Let's see, we doing okay on time? What's our time here? Oh shoot! Okay, I gotta hurry up. What are the parts to the liver? What are the functions? So the liver had quite a bit. It does have lobes. So basically the parts to the liver, you have the liver lobes, you have the gallbladder, and you have the bile duct. Those are, and then you have the hepatic artery and the hepatic, or the portal vein. So you do have, those are the primary structures. The functions of the liver are this slide, which is slide 69, which, material for digestion, synthesize nutrients, excrete toxins, produce proteins and clotting factors. So a lot of important stuff. Where is bile produced? Bile is produced within the liver from a hepatocytes. So if you remember, I talked about a bile salt. I'm going to come back to that in a minute. So bile salt is released, draws water from a hepatocyte. It creates bile fluid that is stored in the gallbladder till it needs to be released through the gallbladder into the duodenum. And it says that's where so bile is produced in the liver, stored in the gallbladder. It says, what is heme? And what is bilirubin? Heme comes from a dead or destructed red blood cell. So when it's lysed or it breaks apart, when the cell dies, the body reabsorbs the amino acids from the cell, the globin from the cell, the iron from the cell, but the heme is the other part of hemoglobin that can't be reused. And so when it goes to the liver, it becomes bilirubin. So heme goes to the liver. It's conjugated into what is called, becomes conjugated bilirubin, which is part of the bile fluid that gets released in the intestine. But if you remember, all the blood flow from the intestine comes back to the liver. So some of that bilirubin gets absorbed back, and then some of it can go back into circulation and is excreted in the kidneys. So the two places the body eliminates bilirubin is through feces and urine. Now, when an animal I said what happens when an animal is jaundiced, and that means the animal has elevated bilirubin. So if it's too high, they become very yellow, gums, mucous membranes, whites of the eye, even their skin. Now the reasons, I told you there was 3 reasons for jaundice, and that's the prehepatic, meaning too many red cells die, intrahepatic, meaning, there could be an actual liver issue, hepatitis, blood flow issue, or what's called posthepatic, which means, the bilirubin is trying to get excreted through the gallbladder or through the bile duct, and maybe there's an obstruction, and then that, since it can't get excreted into the intestine, it gets reabsorbed in the blood, and then it elevates. What is the liver's role in regard to glycogen? So if there's leftover glucose, the body will use glucose, it can be stored in the liver. When it's stored, glucose becomes glycogen. And then if you remember, I said insulin takes glucose from the blood and moves it into cells. So insulin lowers blood glucose because it takes it out of the blood into the cell. If there's low blood glucose, the body can access the stored glycogen. It releases glucagon. I know there's all these words that sound alike. Glucagon is released from the endocrine pancreas, and it will convert stored glycogen into glucose that can raise blood glucose. So they have an opposite relationship. Insulin lowers blood glucose, glucagon raises it. So, and there are places, excess glucose can be stored in the liver and the muscle and the fat. So there's glycogenolysis versus gluconeogenesis, so they're a little bit different there. And I already, I'm not talking a whole lot about the lipid digestion and absorption, carbohydrates. You've already, talked about some of this in nutrition. It says, what is the difference between the dog and the horse cecum? So key difference here is, first of all, is the cecum is almost inconspicuous, almost non-existent in the dog. And the big thing here is the dog's cecum is not essential for digestion. It's like a little pouch, versus the horse has a very large cecum because this is their primary, where they digest their forages. They don't have a rumen, and they eat essentially very close to what a cow does. So this is where they're going to process, so it's essential for digestion in a horse. It's a larger chamber to increase that surface area for digestion and absorption, where in a dog, it's not an essential part of the digestion process. It's kind of like a little inconspicuous pouch. Now, and also here, even though I didn't ask a question, remember, in the horse, it goes ileum, cecum, colon. In the dog, it's ileum, colon. In the cat, ileum, colon. And in the cow, it's ileum, cecum, colon, except that the cecum, they kind of call it a blind, stuff just passes through because it's already fermented and digested from the rumen. So when it passes through their cecum, it's just kind of going through. Where obviously in the horse, any species that it's a hindgut fermenter, they are reliant on their cecum for digestion. What is the function of the pacemaker cells in the large intestine? That's to control the flow of contents. So because contents, this is where in the large intestine, there's a continuation of some digestion, but the majority of the water and ions are absorbed in the large intestine. So the pacemaker cells control the flow so that things don't just go out rapidly. It can spend, like I told you, a practical tip, when an animal is constipated, and what happens is that stool is remaining in the large intestine, it just keeps absorbing water from it, which is not helping the problem. So sometimes when we finally get things to pass, it's very hard, dry stool because it's kind of sucked all the water out. So it's kind of caused a problem. And then what is the primary function of the large intestine? Well, I just said, this is where feces is formed, and this is where a large amount of water is absorbed. And then obviously I didn't ask okay, nothing will go past. Oh, and then at the end here, once stuff moves into the rectum, which is the very, very end, then there's going to be the defecation reflex. So the presence of fecal material at the rectum causes the colon and rectum to contract, but there is an anal sphincter, which the animal does have some control over. I mean, if you leave your dog for 24 hours, your dog is probably going to poop. I mean, eventually, they'll hold it probably as long as they can, but eventually the need to go is going to be so great So they can control that anal sphincter, which is innervated. You know, the colon and rectum contracting are autonomic. So that's like an automatic contraction. It's just the sphincter, they do have some control. Let me go back real quick, show you. So this is the picture to review that's going to be on the test. There are going to be there's not going to be an image of the heart. There's not going to be that image of the head of the dog that was like cut in half, you know, that we labeled for the respiratory system. So just this picture. But you know, anything on this picture is fair game. You've got the liver, gallbladder, the bile duct empties into the duodenum, and then here's your pancreas, and it deposits its digestive enzymes into the duodenum. Now, your spleen and kidney are in this picture. They are not having a digestive function. That's just where they are anatomically. You do have the aorta, which is going to the abdomen, and the vena cava is the blue one here, which is going back. But remember, this portal vein, this is the blood that's coming from the intestine. So remember, all that blood goes from the intestine back into the liver. It can process the nutrients. It can get rid of any wastes. And then after it's gone through the liver, that blood will enter the vena cava. So it's kind of, think of it this way. It's like in a road, the intestinal blood flow, that's got to go back to the heart. The liver is kind of like a stop and, you know, on the way. It's like, well, hey, let's just stop at the liver, process our blood, and then we'll go to the heart. But that's why there's a third vessel here. Normally you have two, like an artery and a vein. This vein is taking that blood flow from the intestines back to the liver. So are there any questions about the picture? I'm going to look and see if I have a Kahoot that I can either try and post the link or my information to your Moodle page that you guys can work on your own, that may have some review questions. You also will have access to your old tests in Teacherese. So you can, you know, especially the blood flow is going to be on there without a word bank this time. So you have to do it just like you did on the respiratory test, but no word bank. But you've done it now twice. So my best advice is to write all those parts down on either index cards, Post-its, whatever, and mix them up and see if you can put it in the correct order. So the blood flow through the heart and the respiratory system will be on there. And does anybody have any questions about either cardiorespiratory or a question about another thing you want to talk about? I know it's okay. Well, I used our whole time. Email me if you have any questions. My computer's being a little slow. Obviously, it's ready to quit on me. I'll get this video converted and I'll put maybe the link. So check Moodle. Happy studying. The midterm will be on Monday. More than likely, it's going to be on Teacherese at this point. And I will email you about the timing of the test. I usually open it at class time, but probably maybe then till like 5 or 6 you'll have time. And I'll get all the homework grades updated. I'm trying to work on that as well. If you don't, oops. Let me do this.